Thank you very much for the invitation today. I really appreciate it. So what I wanted to talk about today are really some of the changes that happen in Alzheimer's disease before you get cognitive impairment. So these are the things that happen decades before you actually start to have someone say, hey, something's wrong with your memory, or I've noticed that you've got some memory issues. And so these are the things that are hopefully preventable, hopefully things that we can target. So I wanted to kind of just tell you about two types of Alzheimer's disease before we start so that you'll understand that we're really talking about two kind of different groups here and how we utilize these groups in research and why sometimes Alzheimer's disease is so hard to study. So late onset begins or is classified as anyone who develops the disease after the age of 65. About 95% of all the cases are late onset. And while there can be a familial component, meaning that if your dad, for example, has the disease, you're more likely to have the disease, the majority of these cases are actually what we call sporadic, meaning that we don't actually have a reason for why you develop this disease. We do know that it's probably a makeup of a whole bunch of genes interacting in a way with the environment, and that's part of the reason why it's so hard to identify. To date, there haven't been mutations that are linked to the sporadic version of this disease that are very easy to identify. Now, if you look at our animal models and primarily what we study, we really actually study the early onset groups. These are individuals that have an onset of less than 65 years of age. They make up less than 5% of the cases, and they actually have a genetic mutation in which there is an autosomal dominant inheritance pattern, meaning that if you have this mutation, you will develop the disease. Now, one of the nice things, or that's not a nice thing, but one of the things that's beneficial from a research perspective is that with somebody with this mutation, if their father, for example, developed the disease at 55 years of age, we kind of know that they're going to develop the disease pretty close to that. Now, why is that useful? Because, how does that affect our ability to do research? Well, the problem with the late onset group is we have no idea when these people are going to develop the disease, nor who will develop the disease. So in terms of doing prevention studies or trying to even look for things like biomarkers, you have to start following them for 20, 30 years to try to figure out, is there anything that predicts who's going to go on to develop this disease? And if you try to intervene, you don't even know if they're going to get the disease. We're not like heart disease. We don't have good biomarkers right now that we can easily identify. You're probably going to have a heart attack eventually based off your cholesterol, for example. We don't have that. So a lot of the research has really started focusing on these early onset individuals because we know if you have this genetic mutation, you'll develop the disease. We have a good idea of approximately the time at which you will develop this disease. So why do we really want to actually study the preclinical phase? Well, if you look at cognitive function across time, eventually with age, everybody's going to develop some cognitive impairment, a little bit of cognitive impairment. But what happens with mild cognitive impairment and dementia is that is sped up. It's happening faster and the decline is much quicker. Unfortunately, if you look at the brains of these individuals, they actually have a lot of neuron loss already, even in MCI, which is mild cognitive impairment. That's problematic because other than stem cells, we really have no way to regenerate these neurons. And even if we regenerate the neurons, theoretically, if stem cells worked perfectly and we weren't having all the problems with them, if it worked perfectly, it might allow you to make new memories, but it's not going to capture those ones that are already lost because of the degeneration process. So really, that's why, as of late, you'll see a lot of focus is more on trying to figure out what's happening before someone develops this disease, trying to target the preclinical phase before there's massive neuronal loss throughout the brain. So I'm going to talk about two things that we do know happen during this preclinical phase and kind of what we're doing in the lab to look at those. So one of the first things that we know happens is a glucose hypometabolism. You might have heard Alzheimer's disease referred to as type 3 diabetes, and there's a reason for that, and that's essentially if you look at the brain, glucose is not able to be utilized the same way it is in a cognitively normal person. And we've known this for about 30 years. We know that if you look at someone with Alzheimer's disease, the brain utilizes glucose to a lesser extent. But for a long time, that was thought to be the case simply because if you have massive neuronal loss within the brain, you're probably not going to have any neurons to utilize glucose. So the decrease in glucose utilization is just probably a function of neuron loss. And that was kind of how we viewed it for a long time. But in recent years, it's been discovered that if you look at those early onset individuals and you start tracking them 20 years ahead of time before they go on to develop the disease, you actually see this glucose hypometabolism about 20 to 30 years prior to them developing the disease. So it's an extremely early thing that seems to happen. Now, we've also since figured out that this is also the case for sporadic. Again, it's much, much harder to study, so there's not as much data out there for that, but it is the case that you can see this decades ahead of time, this decrease in glucose utilization. Now, this is not limited to Alzheimer's disease. It's actually pretty common across most neurodegenerative conditions. It just varies where in the brain you see this decrease in utilization. One of the interesting things we know, and you'll see a lot of lately in the news, is that traumatic brain injuries or multiple concussions, for example, are risk factors for Alzheimer's disease. One of the hypothesis reasons for that is because even with a very mild head injury, the brain cannot utilize glucose properly. And this is not a short-term thing. If you look 3 months later, these individuals still cannot utilize glucose properly. If you look a year later, some of these individuals, particularly the ones who are still exhibiting impairments, they cannot utilize glucose properly. So this has been theorized to be one of the reasons why TBI is a risk factor for Alzheimer's disease. We actually see the same thing in seizures. So seizures, for a long time it was known that you actually will see a huge spike in glucose utilization when you're having a seizure, which makes sense because your neurons are extremely active all at once. All of your neurons are firing, and so you need glucose in order for that to happen. Then people started looking a little bit closer as to what was happening in between the seizures. And the first thing they started noticing was there's actually a decrease in glucose utilization. The brain is not utilizing glucose very well. And then they said, okay, well, what happens if we keep looking further and further back, prior to when someone actually develops a seizure disorder? Can we see this hypometabolism before that? And it turns out, yes, you actually can. So even in seizures, you see a decrease in glucose utilization prior to the onset of a seizure disorder. So all of these things are suggesting that glucose hypometabolism is actually a very common thing across many neurodegenerative conditions. In addition, we know that epilepsy is a risk factor for Alzheimer's disease and a very common comorbid disease. So if you look at late onset Alzheimer's disease or the sporadic cases of Alzheimer's disease, they have a 3-fold increase in seizure incidence compared to the general population. Now, if we look at those early onset individuals, 87-fold higher seizure incidence compared to the general population. And about 30 to 70% are comorbid for a seizure disorder, and it kind of just depends on which mutation you have, whether or not you're 30 versus 70. Now, this increased activity all suggests that this is another important factor, but to date, we really just know that these people have these seizures when they have Alzheimer's disease. But this led people to start looking further back and saying, well, is it possible that there's actually an increase in activity maybe below the seizure threshold? So we're not talking about necessarily having seizures, but could the brain still have increases in activity that might also be another very early change that happens in the preclinical stage? And sure enough, if you look at the second thing that we've tried to target in my lab, is the hyperactivity of the hippocampus. So the hippocampus is the primary learning and memory area of the brain. It's one of the first regions targeted in the disease. It's one of the first regions where you develop some deficits because it is, again, the memory area. So if you look at this region, again, you see a hyperactivity of this region years prior to a diagnosis of Alzheimer's disease. And again, we know this primarily from the early onset because we know when they're going to develop it, and so they've been enrolled in all of these trials where they get followed for years and years and years. But it's also been shown as well in some of the sporadic cases. So this is an example of when a person is performing a memory task, they measure using fMRI the regional activity within a particular brain region. So the ROS is just region of interest, and in this case we're looking at the hippocampus on either the left or the right hand side of the brain. In the control, when they're asked to perform a memory task, you see increased activity within that brain, which is something that should happen because if you're utilizing a brain region, you expect to see an increase in the activity. But when you look at mild cognitive impairment, and we know now even earlier in the preclinical phases, you actually see too much activity. You actually see more activity than the controls. Now you'll notice in the Alzheimer's or the AD over here, there's almost no activity, and that's again because of the massive neuronal loss. There's basically nothing happening when they're trying to perform a memory task because they just really don't have hardly any neurons left within that region. So it turns out that if we focus just on the green and the purple lines here, and we look at what happens to the hippocampal volume, which is basically our neuron loss, it's a measure of neuron loss that doesn't require us to take the brain out, we see that in preclinical stages, there's really no neuron loss. And it's really not even until the later to mid stage of the mild cognitive impairment that we're getting neuronal loss that very quickly declines within Alzheimer's disease. So that's not really our best measure, and that's good that we're not seeing that preclinically because it means that we've not already started to lose the neurons. But what we do see with the hippocampal activity is that it starts to increase, not a ton, but definitely increases a measurable amount during that preclinical phase. That means that that's something that we might eventually be able to use as a biomarker as well as something that we can actually target because it is occurring before the neuronal loss. So the question, one of the questions I commonly get and that we had to answer, of course, in the lab at one point was, is this increased hippocampal activity simply because they're having a compensatory response? Or said another way, does their brain have to work harder because they have a problem? And so is the increase in activity that you see on these things like an fMRI simply due to the fact that they're having to work harder? Well, no. We and others have shown that if you actually decrease the activity, you do decrease the memory deficits. So they can actually perform better in the fMRI if you decrease the hippocampal overactivity. So that's been something that we've continued to target and plan to move forward with. But I want to, before I move kind of forward with where we're going, I want to try to make this clear on why we think that these two are almost like a feed-forward loop. So we know that there are multiple risk factors for the disease: brain injuries, like as I mentioned, TBI, stroke we've studied in the past, hypoxia, diabetes we've studied in the past, vascular dysfunction we're studying now, as well as even viral infections, for example. All of those are known risk factors for the disease. And if you follow down what they do, you can see that they do two things that I've already mentioned, one of which is to reduce glucose utilization and also create these aberrant network functioning, which is basically that hyperactivity within the brain. So if you look at all the things that we consider to be risk factors, we know they increase oxidative stress and neuroinflammation. That then leads to reduced glucose utilization and energy deficiency. You get alterations within the networks and how they communicate, particularly you get too much activity. This then leads to a further energy deficiency, which then leads back to reduced glucose utilization. So it becomes kind of this forward feed loop, which is one of the reasons why it's believed that every time you have some sort of risk factor or insult, it can build upon itself and it can make it worse. And even though it happened 20, 30 years before you ever developed the disease, how does that happen? Well, this is an example of how it can kind of slowly build. So how might we prevent both glucose hypometabolism and neuronal hyperexcitability? Well, in the past, we've tried various drugs. We in particularly like the process of repurposing drugs because they're already FDA approved. They already have toxicity profiles on them. So in the past, we've used things that we knew were safe. Take, for example, Riluzole. It's an FDA approved drug. It's been used in the past to treat ALS, not very successfully, but there are similarities between ALS and Alzheimer's disease, and we thought, well, let's try to see how this might play out in our model. One of the things we know that Riluzole does is it decreases glutamatergic activity, and glutamate is the primary neurotransmitter system that's driving that hyperactivity within the hippocampus, and so this is known to decrease that. In addition, it's also been shown to have to rescue some of the hypometabolism that's observed in disorders like Huntington's disease. So using this, we've been able to demonstrate that it improves cognitive function, it decreases neuronal hippocampal activity, it rescues some of the synaptic alterations that are very common, and it attenuates Alzheimer's pathology. And this is actually currently in a phase 2 clinical trial for patients with mild Alzheimer's disease. And I'm really hoping that this ends up producing some positive effects. I'm kind of concerned that they're testing it on mild Alzheimer's patients instead of maybe more of the preclinical, but hopefully it will have some positive effects. The last one I'll talk about briefly are ketones. So how many of you have heard of a ketogenic diet lately? It's kind of everywhere, right? So there's partly a reason why this is. It's been really pushed lately within the neurodegeneration field because we are seeing hypometabolism across the board for so many neurodegenerative conditions, and it is being recognized as something that's happened very early within these. And as this kind of has been around, it's actually been around for a really long time. It started back in the 1920s as actually a diet that was given to individuals with seizures. So children who do not respond to seizure medications can go on a ketogenic diet and actually see really pronounced effects. It's a little hard diet to follow, but it actually produces very nice effects in these children. So this is basically the idea that if your brain cannot utilize glucose, is there an alternative fuel source that you can provide? And the answer is yes. And historically, if you think back in time, we probably didn't utilize glucose constantly. It's a little naive to think that we constantly were able to go out and eat large sources of carbohydrates. And so from a Paleolithic type standpoint, ketones have been an alternative fuel source from the beginning. I'm not exactly sure why we neglected them for so long, mostly because none of us make them unless you're eating a pretty low-carb diet. But they have been an actual alternative fuel source. And so in the case of the seizures that I mentioned, it was primarily because people noticed that when children had to fast, they actually reduced their seizures. And one of the things that happens when you fast is that your body doesn't have any glucose. When you don't have any glucose, fatty acids get released from fat cells. Those travel to the liver then converts them to ketones, and these ketones can be used as fuel throughout the body. They are known to have very beneficial effects, in part because they are a much cleaner source of fuel than glucose. And so there are several kind of beneficial effects that can be seen from these. They're more energy efficient than glucose. Like I said, they burn a little bit cleaner. They can stimulate mitochondrial biogenesis. This is actually important across almost every neurodegenerative condition. Almost every neurodegenerative condition is associated with a loss of mitochondria. So it's actually quite beneficial to increase mitochondrial across the board, pretty much. Pretty much everybody could stand to have more mitochondria. It also protects against glutamate-mediated apoptosis. So remember the thing I mentioned previously with the hyperactivity, that's mediated by glutamate. So if you can stop the downstream effects of this hyperactivity, that's quite good. And so it can actually prevent the cell death. It can also enhance the conversion of glutamate to GABA. So does everybody kind of know what GABA is? Probably not. Well, GABA is our major inhibitory neurotransmitter of the brain. If you take something like Xanax, for example, that's something that activates GABA and decreases the activity. Oh, actually, if you just drink alcohol. So if you drink alcohol, that leads to an increase in GABA and depresses the activity of the brain. So that's very beneficial if you have an issue of hyperactivity within the brain. You want to kind of suppress that, which is why it's so beneficial within seizures and things like Alzheimer's disease. Very anti-inflammatory. Decreases cell death from apoptosis, increases cerebral blood flow. So this can be important for things like vascular dementia, which is another form of dementia we're currently starting to look at more. And it also decreases damage from TBI. So this has been commonly used. DOD is very interested in this, in exogenous ketones, because of the potential benefits for TBI, as well as diving. Diving produces a hypoxia that they're very interested in reducing. So those are the two major things that we're currently studying in the lab, as well as some other stuff, but I want to thank my lab members and the funding and take any questions.